Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base biotech podcast, and this is number 53, and it's going live on April the 3rd. It happens to be Good Friday today, which is, of course, a holiday in some places and not others. One thing you can say in this part of the world is it's probably raining. Scotland is wet at the best of times, but 2 weeks of rain to cover the entire school holiday is a bit much. Not that you can file an official complaint. Last week we chatted with Hans Eriksson, Chief Medical Officer of HMNC Brain Health, about ketamine and treatment-resistant depression, and with Krishna Polu, CEO, President and co-founder of new kidney health biotech company R1 Therapeutics. We managed to get that one in just in time for Kidney Month, and this week we're only one day out with Autism Day, which was yesterday, but we're still going to talk about it, with Manish Arora, CEO of LinusBio. And a remnant of last month's kidney theme, we chat with Thomas Kledal, CEO of Synklino, about improving kidney transplantation outcomes through better cytomegalovirus prevention. If you do have news or suggestions for who should be on the podcast, or indeed any other suggestions or comments, please do get in touch at jim@deeptechdigest.com, which is also the address you can use if you're interested in advertising with us, sponsoring a podcast episode, or even for event coverage. First, it's time to go through some of the headlines of the articles you may not have read over at deeptechdigest.com. Crossbow Therapeutics has raised $77 million to treat cancer. The FDA has approved a pill for plaque psoriasis, and a Lyme disease vaccine has showed strong efficacy in a phase 3 trial. A copper overload kills cancer cells. Researchers believe they are moving closer to preventing pandemics. Radiotherapy may help reduce the risk of lymphoedema in patients with breast cancer. Tumor DNA in the blood after pre-surgery treatments predicts if breast cancer will return. And precision medicine helps more patients receive genetic diagnoses. The search to diagnose rare disease is changing with a new AI breakthrough. The world's largest clinically linked spatial biology atlas has been launched, and a bispecific ADC has received IND approval. You're listening to Acronym Radio. Not really. And you can read all of these and plenty more at deeptechdigest.com. So to the interviews. First this week, because yesterday was World Autism Day, we have a conversation with Manish Arora, CEO of LinusBio. It's a company that has developed a test that can rule out if someone has autism. And it's a story that is very personal for Manish. All right, hi Manish, thank you for taking the time to chat with us today. First, could you tell us a bit about LinusBio?
I'm the founder and CEO of LinusBio. I'm also professor and vice chairman of environmental medicine at the Mount Sinai School of Medicine in New York in the US. The company is a spin out of my academic center, and what we do is that we have developed a platform that looks at all the non-genomic factors, at exposomes. It's a relatively new term. What it means is once you move above your static genetic sequence, there's a lot happening in your body and outside your body. So it's how your environment impacts your health, but also how your body reacts to that environment. Our key innovation is that we can generate data that's like taking 1,000 samples of blood every hour. So if I were to come to you and say, Jim, I'm going to prick you with a needle every hour for a whole month, you don't get any sleep and you run out of fingers for me to prick very quickly. But I collect these drops of blood and analyze them. What we figured out was to achieve this impossible sounding task, we could do it with just one strand of hair. Why can we do that? Well, it turns out that biology has given us this gift that inside hair, you can imagine them as growth rings because hair grows incrementally. So there's a growth ring every hour in a sense, just like a ring in a tree is every year. Getting into a hair is not easy. Some hair is very curly, some hair is thin, some is thick. So we started building robots because human hands couldn't manage that. So our robots take a hair, make it perfectly straight, and can open it up like a book, lengthwise. Instead of cutting it cross-sectionally as you would do in a tree, we are sort of slicing the tree from root to tip, which is very difficult, but again, robots can do that. And inside, we take molecules along every growth ring, and since the growth ring is every hour, it's like taking a blood sample every hour. And human hair grows about 1 centimeter a month. So for most individuals, we just get a single strand of hair and analyze it. I'm sure your fans and your audience know that there's not a lot of hair left on your head, and I'm bald too. So there's only one exclusion criteria, and unfortunately, Jim, you and I are the exclusion criteria. That's the core technology. Now we choose to apply this technology not to develop cosmetics or shampoos or things like that, but to impact areas of health and disease where there is immense public health need, because that's what I do. I'm a professor of public health in a sense and environmental medicine.
So we can't be helped with no hair, then we're beyond.
I am constantly watching for innovations at other centers who have figured out the cure to baldness. I do miss my hair. So until that day, we can't be helped. But I'm sure someone will figure out how to regrow our hair.
So when it comes to ASD, I wonder if you have any statistics on that and what the current methods of diagnosis are?
Absolutely. So one of the areas we chose to impact was neurological disorders, and among them a prominent one is autism spectrum disorder, or ASD. There are both professional and personal reasons why I chose to work in autism. One is that professionally I was surprised that when you come to most neurological disorders, the clinical practice is so different to the way other areas of medicine work. Back in the day when I was a clinician, I would do some surgical work in the mouth, in the oral surgery field. Before I even touched a patient with a scalpel, we would have a detailed path report. For example, is this person a hemophiliac? Are they going to bleed on my table the moment I touch them with a scalpel? Or if they have undiagnosed and uncontrolled diabetes, will they struggle to heal and I'll have problem with wound healing? Things like that. In autism, we fill out a questionnaire. These diagnoses are made based on questionnaire tools. These are good questionnaires. I'm not criticizing them. But for me, these questionnaires should be the beginning of the story and not the middle and the end. There is no biochemical testing. Yes, this is a spectrum disorder. However, it is a true biomedical condition. So we decided, because genetics hasn't given us a good diagnostic tool for autism, we should try and do that. And that's what we exactly did. It took us almost 10 years. We did studies around the world in Sweden, looking at identical twins and Japanese babies, 1,000 families in California, poor communities in Mexico, and so on. And we found a biochemical marker using this technology of analyzing growth rings in a single strand of hair. And then we took this technology to get FDA breakthrough designation and several other forms of diligence. In the US, we have a system called CLIA, we have the international standards of ISO, and we have all of those diligence. So now we are helping families by using this test to detect the risk of autism early. If I may continue for just another minute to describe why the early detection matters in autism. Beyond this very subjective diagnosis in autism, what surprised me was that for a condition that's heritable, that means it's present at conception. So even before you're born, you carry the risk factors for autism, and it's certainly present at birth. The median age of diagnosis has been stubbornly stuck around 4 to 5 years of age in the US, and in Europe it's even older. In Japan, it's later in life. So what's happening here is a very sad sort of convergence of problems. One is our brain is really developing rapidly in the first year of life. That's why in the second year of life, children have language and speech, because they have been picking up language right from early life, even prenatally, some researchers would say. Yet we deliver therapy at 4 to 5. So we are missing that very critical window of opportunity when delivering therapy would have an immense positive impact on an autistic child's future. And research has shown that it does. It is, without fair early intervention, is very beneficial in autism. What was missing was an objective marker or objective tool to detect autism early in life. And that's why we said, hey, if we can detect it early enough, children can get therapy and everyone is better off. Even in the US, we have this unusual system of private health cover for everybody. You have to buy your own health cover. We are very envious of the British NHS. Delivering therapy too late doesn't benefit the child, costs more to the healthcare system, whereas delivering therapy early is a win-win. The child is better off and it will cost less to the healthcare system.
What are the challenges with diagnosing autism or previous to what you've been able to achieve?
Because the diagnostic standards currently are based on observation, essentially you have to develop enough symptoms, enough behavioral symptoms for a clinician to be able to say, yes, you behave autistic, therefore you are autistic. Now, again, let's compare it to other aspects of medicine. You wouldn't accept that for diabetes, would you? A doctor couldn't walk up to you and say, well, I think you look like a diabetic, therefore you are. Or in cancer, you look like a cancer patient, so therefore you are. Yet in many of these conditions, there is no biology in the very definition. The biology or the definition of autism is entirely behavioral, entirely observational. And so what happens is this creates a perfect storm. The diagnosis can only be made when the child shows enough severity. Often the parents are noticing something very early in life, like 6 months or so. Yet there are no tools because you really can't objectively identify a child's behavior when they're just a baby. The second thing is these behavioral assessments take a very long time. I, as a parent of a neurodiverse child, have experienced that. One assessment for my daughter was 6 hours long. And so no child can sit for 6 hours at the age of 6 or 7. So we would travel 2 hours from where I am, book into a hotel and do it over 2 or sometimes 3 days. So this became a whole event. And then the wait lists for diagnosis are huge. In so many centers, they're over a year long. If you really call your doctor with a concern for your child today, you'll get an appointment in 2027, a year later, which is just unacceptable in this day and age. Also, there's a national and I would say global shortage of developmental pediatricians, the specialists who can diagnose autism. And going back to the US eclectic healthcare system of private insurance, the reimbursements for diagnosis are so low that it is not an attractive field for young graduating doctors to get into. So not only do we have these long wait lists because it takes so long, we have a shortage of doctors actually training to become developmental pediatricians because there is no incentive there. Wouldn't the world be a much better place if we had a proper biochemical screening tool where if you have a concern, you just cut some hair at home while the child is sleeping, and rather than wait a year, you get an answer in under a month. And if the answer is no, you're not on the autism spectrum, you don't have to join that wait list. So the other sad reality is if you go to a hospital wait list for autism, sometimes 50 to 70% of those kids are not autistic. Population prevalence is 3%. So 70% of those kids are actually in the wrong wait list. They're going to wait a year and then get to the front of the line and then find out, oh no, you're in the wrong queue. You have something else that could have been addressed more easily. Go join a different queue. The solution to this perfect storm is a very simple idea. Have a screening tool very early in life. So ClearStrand ASD, that's the name of the test that we have developed, is a rule-out test. It's meant to screen very early in life, and our regulatory approval allows us to screen as early as 1 month. Why 1 month? Well, some babies are not born with much hair, but by 1 month, they all can give me a centimeter of single strand of hair. So from 1 month onwards, up to the age of 10, we can rule out autism with over 95% accuracy.
You mentioned about the long lines of people waiting for tests and the fact that many people don't have autism. What are the conditions that could be mistaken for autism?
And again, I'll share my own personal journey. Like I said, I have a neurodiverse daughter. Turns out she has a very high level of ADHD and a very high level of anxiety, and shows some perseverance behavior, which is like sort of repetitive. She likes to do a few things quite often. So often, doctors would say, he thinks she's autistic. However, having a correct diagnosis, even between these conditions that can mimic each other and can be clinically confusing, the difference is enormous. And again, I'll share my personal example. ADHD and anxiety, there are medications to treat those. And the moment I started my daughter on those medications, it's a life-changing event. It's a different person I'm dealing with. I could not imagine my child would ever stand up on stage and give a 20-minute presentation on Neil Armstrong. And that's what she's doing now. But when we did not know that she has these ADHD and anxiety, and we thought she was autistic, we were like, well, there's no medication, right? There's no approved FDA drug for autism. So we kept throwing therapy at her, and yes, certain things were improving her language. She was very comfortable with her therapist. But the moment she went outside and met a stranger, the ADHD and anxiety kicked in, and in school, she's a troubled child. So I'm sharing a very personal anecdote with your audience because the conditions that often get confused with autism are ADHD, anxiety, and then some general developmental delays. But knowing that that is not real, your child is not really autistic and needs different care can be a life-altering event, like in my example. Treat for ADHD, treat for anxiety, and my daughter now is literally a different person.
Right, so could you maybe tell me a little bit about how your ClearStrand test works?
ClearStrand ASD, as the name suggests, the strand is from a strand of air, and clear because we want to rule out. By design, we wanted this to be a tool that strengthens the relationship between the doctor and the patient. So we are not trying to replace any clinical expertise at all. I always told my team, we're not trying to develop something like a pregnancy test that tells you yes or no in the isolation without a doctor. Instead, we're more like a cholesterol test. It tells you, gives you a very important piece of information, which your doctor will use to make a decision for you. It's a rule out test. What that means is the test will deliver an answer saying you're not on the autism spectrum, and that is, we're very accurate there, about 95% or higher accuracy. When we cannot rule out, we say, no, you need to go speak to the doctor because we have detected the biomarker. That doesn't mean you definitely are autistic, but your doctor needs to make that decision. Just like if you have high cholesterol, it doesn't mean that you necessarily have heart disease. But your doctor will guide you on what next steps come. This came after discussion with dozens of doctors of different kinds. Pediatricians, for example, told us, hey, when a family comes to us and they're concerned about autism, we have no really reliable tool. So we just keep referring them to a specialist center, knowing that the wait list is a year long. We would love a tool that allows us to say with confidence, no, you're not on the spectrum. You don't need to go to the autism specialist waitlist. You probably have something else, maybe like a language delay. Let's talk about ADHD or something else. He also told us, as a general pediatrician, I'm not a specialist in autism. So for me to rule it in, this is not the place. I have, 20-minute appointments, and, I have not been fully trained to treat autism. So I want to send people to the specialists who I know have a high likelihood of getting an autism diagnosis, but not just refer everybody. So this became a very simple tool. When hospitals have large wait lists, we say, give us all your wait list and we will triage the patients for you and say, hey, see these first, they're at higher risk. And these folks, take them off your wait list and send them back to your general developmental clinicians. So again, all of this 20 years of my science has come down to just assisting clinicians and families by saying, you belong here and you don't belong here.
Do you need the whole thing or just the shaft?
Very simple. And I always tell people, if you're spending more than 30 seconds on collecting the hair, you're overthinking it. We send a kit which just contains a simple tube, and we tell them with just household scissors, and your child could be sleeping. If your child has profound autism, they might not like this sudden, parents are coming at me with the scissors. Even my neurotypical children wouldn't want to let me cut their hair when they were little. So child is sleeping, you just clip some hair from the back of their head, and that's it. We only need one strand, but you don't need to worry about just cutting one strand. Just cut whatever you grab in your finger, because gathering one strand is actually quite difficult. So you cut 4 or 5 strands, put it in a tube, send it back to us. You can order it from our website. We actually provide in the price of the test a 15-minute consult in the beginning with an independent pediatrician who has nothing to do with the company. So you get an independent opinion. After the test is performed, you get another 15-minute appointment with the same clinician, again, an independent clinician. So you are getting independent advice along with the results of the test. The test results, we deliver them in 15 days. So rather than waiting months and years, you get an answer within 15 days. And it's sent again to this independent clinician, or you can use your own clinician. As long as they prescribe it, you can get it through your own clinician.
Can it be prescribed by a physician, or can they get reimbursement?
It is by prescription only. Any clinician can prescribe it for their patient, and we just ship out the kit to the patient's address. Given the complicated system in the US, the price is $499, essentially $500. You can use your HSA dollars. So for your non-US audience, what that means is we are allowed to contribute some money towards our healthcare with pre-tax dollars. So whatever your tax bracket is, minus 40%, for example, I'm essentially getting a 40% discount by using pre-tax dollars. It is not covered by private insurance yet. We are working towards that. But like any novel technology, now that we have passed through all the diligence steps, we'll get there. I think we are, about a year away from being covered by insurance. We kept the price purposely quite low compared to the traditional diagnosis. For example, if I wanted to not wait a year and just pay out of pocket for a full autism diagnosis, in my own experience, I paid more than $5,000. So this is 1/10 the price, and it's much faster. And if you use your HSA dollars, it's less than 1/10 the price. So we purposely kept the price manageable, but I want to be fair. Whether it's $300 or $500, that is still a fair amount of money for many families in the US. So we're actively working to getting insurance coverage.
And how about globally? Is there a global capacity for this?
Yes, absolutely. So we are rapidly approaching partners. We are already active in Japan. We have a partnership there, and they use our test in Japan. And we hope to be present in the Middle East soon. And in fact, we have been talking to folks in the UK because there are very long wait lists in the UK. One example was a family that flew in from the UK to the US so that they could legally buy our test. So for them, because it's such a difficult situation where you can't get a diagnosis. If you don't have a diagnosis, you can't get the right therapy. They said, look, we'll just come to the US, get the test done. And, now their child is getting therapy, and they've saved, more than a year, because the wait list where they were was longer than a year under the NHS system.
And is there significance to earlier diagnosis?
It can be life-altering. One of my biggest regrets is that it took us so many years to get the right diagnosis for my daughter, because the brain is really malleable in the first 1 to 2 years of life. There's always some neuroplasticity, even at our age. But early in life, children can pick up social cues, language, and other such essential developmental skills. If we are delivering therapy too late, we are missing that critical window of opportunity. I often describe it in very simplistic, almost silly terms. I say, like, if delivering therapy too late, it's like putting on your helmet after your motorbike accident. The critical window of opportunity is while you're tumbling off your motorbike and hitting the road. If you have a helmet on, then it saves your head. And I'm talking from personal experience, having been young and reckless and been in a motorcycle accident, which was entirely my own fault. Give therapy early. I am such a big believer in giving therapy early in the first 18 months of life, because I've seen the impact on children. They go from low functioning to high functioning. That means they can now hold down a job. They can enjoy social interaction. They can have relationships. They can interact through sport and other means, versus just being able to be surrounded by your own family and not really dealing with the outside world. For any parent, that's all we really want, right? Our child to experience happiness and the good things. We don't want, we're not necessarily concerned that our child becomes an astronaut or not. We just want them to be happy. And that's what early intervention can achieve, taking you to a higher level of experiencing joy.
Recently, the age range of the test was expanded. What does that mean?
Absolutely. So when we first started, it was from 1 month to 4 years of age. As we collect more data, we submit it for regulatory approvals and everything. So now it's from 1 month to 10 years of age. We will keep expanding that. This age range came from a very large study that we did on almost 1,700 families globally. There were 1,000 families in California, some in New York. We also studied a very poor community in Mexico, twins in Sweden, a large representative population in Japan. So we do these global studies that undergo peer review. And then we submit it to the US governmental system, what we call CLIA, and that then becomes a laboratory developed test. So yes, our age range is now from 1 month to 10 years.
Is there a big value to the amount of data that you are able to collect now that you're collecting more data?
Absolutely. We want our tests to eventually not just say yes or no that you're on the spectrum or not. We already have data at a research level that shows we can distinguish autism from ADHD. And as we collect more data there, we will be able to release a test that says, hey, you have neither, you have one of these, or you could have both because the conditions can coexist. And we're also working on what type of autism, what type of behaviors and challenges does this autistic individual face? Whether they will have high levels of anxiety, or they'll go on to develop epilepsy, or will they undergo a regression event. So we are also working on that subtyping of autism because it's a spectrum disorder. It's a complex mixture of symptoms. Another area that's very interesting is that, especially in girls and some boys, we see that symptoms suddenly spike during puberty. Why is that happening? We already know that autistic girls and autistic boys seem to have a different experience on the spectrum. But especially around puberty and for girls, it appears in some cases to become very profound. So we're trying to understand that. So yes, all of this data has immense value because eventually we can help patients with their individualized treatment. That is the goal of precision personalized medicine.
And especially with ASD, in the definition of it, it's a spectrum. Are you anticipating the ability to be able to help in terms of where people are on that spectrum?
Absolutely. I think one of the problems with the autism spectrum disorder is that without a biological definition, it's so wide, you end up getting one label. And I've seen children with profound autism that need 24/7 care, and then very high functioning autistic individuals that have some skills that are well above what a regular non-autistic individual would have, and they're celebrated. We keep hearing of these famous scientists like maybe Einstein was autistic and things like that, or maybe Mozart was autistic. However, what is needed is that we break up this disorder into subtypes. So eventually the answer will be you have type A and another person has type X of autism. But for that, we need biology. It cannot be done with questionnaire. So the ultimate vision that I have is that we need to redefine autism and bring biology into the very definition of autism. And then we can start developing treatments that are specific to where you are on the spectrum.
Do you see any sense that that is potentially going to happen? Do you have discussions with anybody that can take that in that direction?
Absolutely. 100%. We're talking to pharma companies too, and we've already been part of a phase 2 trial for an autism drug. Ourselves, we are seeing data, which in our own data, we are seeing clusters using our artificial intelligence systems that allow us to say, oh, there are differences happening. You have the social interaction kind of, or you belong in the social interaction kind of cluster, whereas you are more in the language cluster. So already we are seeing this, and this is entirely driven by the biological signals. We're not using any questionnaire data at all. Beyond your age and gender, we don't ask anything. So we are really relying deeply on biology. Just like a diabetes test will give you an answer without asking, what's your ethnicity? It will give you a fairly reliable answer. That is the ultimate vision, that we can bring that level of precision to neuroscience.
Obviously, this is a huge project in itself, but are there any other tests that you're developing?
We are close to finalizing a test for Lou Gehrig's disease, or amyotrophic lateral sclerosis, ALS, as it's known outside the US. This is a terrible condition, a neurodegenerative condition that happens mostly later in life, but I've seen patients in their 30s and 40s, but mostly it's 60s and older. And by the time you are diagnosed, your symptoms are quite advanced, and the average lifespan after that is 5 years, but the quality of life deteriorates very quickly, very rapidly, and it's really a terrible condition. Again, it's a condition that's diagnosed largely on observation. There are no real good biomarkers that can detect ALS well before any symptoms are present. So we're working on that. And the other area we are hopefully in the future we will have an impact is on GI disorders. Again, for many GI disorders, we don't have a good biochemical test. I had that sort of epiphany when I was lying on my doctor's table getting ready for a colonoscopy, and I just thought, my great-grandkids are going to laugh at this whole process when I describe it to them, that someone puts a tube with a camera inside you and then takes a lot of pictures like a tourist in a new place and said, oh, this looks interesting. I'll grab a souvenir here and a souvenir there. And then they make a subjective diagnosis on a scale. I said, why am I lying here? Why did I just drink that, a liter of Gatorade the day before? And the answer is, yeah, well, we need to do a lot better. Our colleagues in endocrinology, they're measuring every kind of hormone, every kind of electrolyte in your body and relying heavily on biochemical tests. Our colleagues in neuroscience in GI disorders are looking at images or questionnaires. We can do a lot better than that.
Would you be able to use your test to diagnose before symptoms even appear?
The hair biomarker, we see that the earliest in our data set is 2.8 years, which is small still. I'd rather it be 10 years. So we're already seeing that signatures exist several years before diagnosis. The second thing there is that now that we have a biochemical test, we can start working on modifying those pathways. Everything we find is modifiable because it's a biochemical signal. If we give drugs, those drugs can modify those pathways. So we want to go from a condition where we're just treating the symptoms to a way where we can treat the underlying pathology. I always give type 2 diabetes as an example of success. There's no cure. If you're diabetic, you're diabetic. But the quality of life, because we've understood the biology, has improved to the point where your quality of life is so similar to a non-diabetic person that other than that sensor that you're wearing on your arm, sometimes you wouldn't be able to tell this person's walking down the street that they're a diabetic. Gone from the time when type 2 diabetes was the main cause for amputations in some countries to now the Tour de France, there is a team that everyone is a type 2 diabetic. In fact, to be on that team, you have to be a type 2 diabetic, and each and every one of them is way fitter than I am. So why can we not achieve that for autism or Lou Gehrig's disease or IBD? I think the answer is that we just have moved away from the fundamental principle that if this is a disorder, we must define it using biology.
You mentioned AI, and how has that and how will that continue to help your test evolve?
We generate from a single strand of hair up to 10 million biochemical data points. So it becomes a very large amount of data that traditional statistics cannot handle, and human eyes can no longer see patterns ourselves in such a massive data matrix. So our AI tools have now become so powerful that they very quickly can say, oh yes, we can cluster these separately. In fact, our AI models not only clusters autistic individuals and not on autistic individuals, it also works differently if you are a male autistic individual versus if you're female and autistic. So it's already picking up that biological difference between the genders. So on the ClearStrand ASD, the autism model for boy and girls actually runs differently because the AI just knows, oh, this is a girl autism case and this is a boy autism case. And without AI, we would not be able to do it. We are also developing, along with this other tool, because we measure so many environmental exposures, you'll be able to tell whether you're autistic or not. Hey, this is your life's environmental exposure history. This is your load. But how do we guide you once we have all this data? So we are developing AI agents that will talk to you about your data, your metabolism, your environmental exposures. And that itself has relevance to individuals who are autistic because by controlling your environment, you can lessen some of the symptoms. Like many of our anxiety and attention issues are linked to exposure to toxic chemicals, for example. By reducing them, you are going to have some improvements on your general behavior and neurological health.
Will you be able to find perhaps connections that weren't necessarily known about before, in terms of there might be one environmental toxin that worsens autism in some people but not others? Are you looking to find that kind of connection?
Many of my very established colleagues in environmental medicine and environmental health have already shown that exposures to several classes of toxic chemicals will worsen autistic traits, whether it's attention, anxiety, social interaction, executive functioning, things like that. there is no one single gene or one single chemical that is directly going to, take a person from non-autistic to being on the spectrum. But the whole mixture together of so many thousands of chemicals that we are exposed to does make things worse. Our ultimate hope is that we can identify which cluster is the main driver for a person and start reducing that to start improving their symptoms. Coming back to the type 2 diabetes example, by modifying certain pathways, you're still diabetic, but you are so high functioning and the quality of life improves drastically. We're not there yet, to be perfectly honest, but that is the vision, to help improve the lives of autistic individuals by using the exposome as a concept to a point where they have a quality of life that is very similar to a neurotypical person.
It's great that there are people who are prepared to discuss conditions close to their heart and determined to tackle them. And what a difference those early diagnoses can make. Next, even though National Kidney Month has just finished, we're talking, at least a little bit, about kidney transplantation, although this next company is tackling more than just kidneys. Thomas Kledal is CEO of Synklino, a company improving kidney and other organ transplantation outcomes through better cytomegalovirus prevention. Hi Thomas, really appreciate you being here for the podcast, and I wonder if first you could tell us a bit about the company.
Synklino is a Danish biotech. It's located in Copenhagen. It's a small team, but very skilled, efficient team. We were founded back in 2019 out of a BII investment, the Bioinnovation Institute. They gave us the first funding to get going. And we have then since been working our way through the early development of the drug candidates, the selection of that, the whole preclinical characterization and development aspects and talks and safety and so forth. So that's taken us to where we are today, just starting clinical development now.
So in terms of what you're working on, I wonder if you could give me a bit of background on CMV and the danger with respect to kidneys and transplants.
Yeah, absolutely. So we are indeed in transplantation and infectious diseases. So it's sort of in that combined space. The challenge is that once you receive an organ, a kidney or other organs, you suppress the immune system for the recipient so that you do not reject the organ that you have just received often after a long disease or dialysis for years. So finally you get this organ and you really don't want to reject it. So you suppress the immune system. So the challenge is that when you suppress the immune system, you can of course not deal with pathogens the same way as you could with a healthy, well-functioning immune system. And the problem here is that CMV is a ubiquitous virus. So most of us will have it. In the cold Nordic regions, maybe it's like half of us will have the virus. If we go further south to Spain or France, more and more will have it. Sort of in the kissing cultures, they have it, all of them. In the cold Scandinavian countries, only half of us will have it. So it's a very frequent virus, and it doesn't really cause any problems if your immune system is well functioning. It's only until that you get immunocompromised that you can no longer control the virus. And the challenge is that CMV or cytomegalovirus, it's a herpes virus. And like all other herpes viruses, it is a virus that once you get it, you have it for life. And the reason for that is that it has this latent stage of infection that you can't really do anything about. And it is then the reactivation from this latent stage that happens after transplantation that really causes the problems for the kidney transplant recipients. And the challenges are numerous. There can be sort of direct challenges for the organ that you just transplanted, and you really don't want to lose that organ. So increased risk of delayed graft function or chronic graft loss, even rejection over time. So that is directly for the organ. Another sort of kidney-related challenge is talks with current antibiotic therapy. So often you provide Valcyte or valganciclovir as a prophylactic therapy after transplantation, and that can actually cause some significant toxicity to the organs. It's a challenging space. besides to the organ, the virus is all over the place. So it can, of course, also cause challenges all other organs in your body, intestinal system, liver, lung, and so forth. It's a nasty virus.
And how is it currently treated?
So the current strategies is for preventing the viral replication to take place is that you provide antiviral prophylaxis after transplantation. So it could be with Valcyte or valganciclovir, as I just mentioned, or newer products like Prevacid or Leptensity. So you provide this prophylactic therapy for 3, 6, and up to 12 months until that you believe that your immune system has recovered so much that it can sort of manage by its own. So that's what you do now. The challenge here is that despite standard of care prophylaxis, still 30 to 40% of the patients that receive a CMV infected organ, which is most patients, they will experience the reactivation. Half of those will go into have disease-related symptoms and these challenges that I mentioned in terms of challenges for the organ or for the whole patient. So that's what you do, and it's not perfect. And while it, for most people, work part of the time, as soon as you stop this, the virus has a tendency to come up again. So it's, as some of our KOLs said, it's like kicking the can down the road. You really just push it ahead of you because none of the current therapies can do anything to this latent reservoir. They can only prevent the virus from replicating once it has been activated. So you will never really get rid of the core of the problem with current therapies.
Because of those challenges, is it something that many companies are working on?
No, it's not. You had the 2 latest drugs in the market. That is one out of Takeda, that's Leptensity, and you had Prevacid out of MSD. They bought that out of a German company, and then they did the final phase 3 development and put that on the market. So those 2 are the last that was developed. There's been a lot of attention to this over the years and a lot of failures. Many companies have tried vaccine strategies, they've tried antibody strategies, and they all have failed. I think it's hard to understand why you would expect a vaccine to really work, because you are removing the immune system in a situation of transplant anyway. And in most cases, or in many cases, the patient already have a very significant immune response against the virus. So vaccinating has, at least for now, showed not to really work. So Moderna, they have just finalized a phase 3 study for CMV, but not in transplant. That is for congenital CMV, which is also a huge problem. But they didn't really reach their endpoints. So I think they are trying to figure out what to do here. It's a very challenging virus to work with. It's a very smart virus. And antibodies, they have often just tried to target major antigens blocking the viral spread, but is a very cell-associated virus with cell-to-cell spread and so forth. So that strategy also seems not to really work. So you really need to think carefully about it when you set out to venture into developing something against CMV infection.
Does the fact that companies have tried to do something and failed, does that influence other companies' decisions as to whether or not to enter a field like this if they see lots of failures?
I don't think so. I think that's not what we meet, at least. I think we meet genuine interest in what we do when we come out there, and that the companies appreciate that what we present to them is novel and is an approach that could potentially go in and change the way we look at cytomegalovirus, because we can not only deal with the virus once it's replicating like the other ones, we can actually target and eliminate the latently infected reservoir. And that is really where we can provide a new therapeutic paradigm. So I think we are met with actually curiosity when we go out and present what we have here.
Speaking of which, then, could you tell me a bit about your treatment and how that works?
I'll be very happy to do that, of course. Yeah. So we have developed a drug candidate, SYN002. And just very high level, what we do is that we treat the organs ex vivo. That means before they are transplanted. They are procured, we put them in machine perfusion, and then we add the therapy SYN002 there for a few hours. We wash, and then you transplant. this is basically what we do. So the other important thing is, as I also mentioned, that we can target the latent reservoir, because the virus is latent in those organs. So if you want to have some kind of impact and effect here, you need to be able to target the latent reservoir in order to be able to treat organs before transplantation. So those things combined give us this novel approach of cleaning basically the organs. So you can transplant a cleaned organ and have better outcomes of transplantation for the recipient. That's really the goal of what we do. More detailed on the drug candidate, it's a biopharmaceutical. It's a single polypeptide, very selective, very efficient. It targets a virus membrane receptor whose function is to sort of internalize immune defense molecules, and we are exploiting that. So we get a very efficient internalization only into the infected cells. And once it's internalized, it's cleaved and activated and is then driving the infected cell to undergo apoptosis. It's simple to manufacture. It's E. coli process and all that kind of stuff. So it's a relatively efficient and straightforward molecule to work with.
What are the challenges that you face when you're dealing with CMV ex vivo as opposed to in vivo?
Yeah, I think at this stage, from corporate perspective, so I think we have to do more teaching, more education of the people that we speak to, no matter how smart they are. This is a novel concept, and novel is great, but it's also challenging. It comes with that challenge that we need to tell people what is latency, what is ex vivo therapy, and so forth. So I think that's the probably the challenge we have right now, and it's also from a market perspective a relatively new market. So we also, of course, have that we need to address and be able to explain how this can constitute an attractive market. And I think we can do that because we are addressing a significant medical need, and there's no doubt about that. And that, I think, will drive the market. On sort of the more technical side, most transplant centers, OPOs and so forth in the US, they already have these machines. They know how to use them. So organ perfusion or machine perfusion is not something that we have invented. Last time I looked, approximately, I think it was 37% of all kidneys in the US, they are going through machine perfusion in order to rehabilitate them. Right now, it's especially used sort of marginal kidneys where you need extra assessment or you need to see if you can sort of improve them. And that is a field that is growing quite a lot right now. You can also see that in England, where they have just implemented these assessment and set up the ARCs, assessment and conditioning centers. They have made 2 right now. They will plan to make 15. So these are centers that are focused on rehabilitating organs, assessing organs in 15 regions in the UK. So you will have these specialized labs who's actually, or clinical setups, who's actually getting the organ, rehabilitating them, and then sending them back to really improve the number of transplantable organs. So it's really something that is growing quite dramatically right now. And I'm really happy to see it was just last week out that Cambridge got this set up here as one of the 2 new centers for this in the UK.
Could you tell me a bit about your preclinical studies and the data that came from that?
Yeah, we have done a lot preclinically, and we have done, of course, all the pharmacology and tox and so forth that you need to do to get to where we are right now. What I think is special about the area that we are in is that we have had the opportunity to do preclinical work in the real target, in the real setup. So it's not only animals, but actually we have done preclinical work in human organs, targeting the virus that came with that human organ, exactly like we are now doing in the phase 1 study. We come in with very nice data where we can show, for instance, in human kidneys that were donated for transplantation, but they have then failed some quality parameter, and with consent, we have then been able to, together with Cambridge University, to study SYN002 in these organs. There we can see that when we take these organs and perfuse them for, in this case, 4 hours is enough with SYN002, we eliminate and then measure the latent reservoir in the organs before and after perfusion, we can see a 95% reduction in the latent reservoir. So a very fortunate place to be in that we can actually monitor efficacy in sort of the real target. So there's little translational risk left starting the clinical study. We have done a similar study in Toronto, UH in Toronto, on human lungs. So we can also show the same effect data and safety data. We have studied all kinds of safety parameters, thorough histology, biomarkers, inflammatory markers, and physiology, and so forth. And so that's really a nice data set to have when you then enter the clinic. It gives you sort of hope that we can do this. So I think that has been a quite unique situation to be in for us with this drug.
And so where does that put you on the clinical pathway?
We are going to start the clinical development now. We have an open CTA in Canada. We actually also have one in UK. So we have had some nice regulatory interactions over the last year. Great alignment in both our data and plans for the clinical study. We are just finalizing the last preparations at the site in Toronto. And then we are starting the recruitment now. So anytime in the coming weeks, we should be up running. Looking ahead is always dangerous, but I expect enrollment here in the coming months to begin. Absolutely, that's where we are, and everybody is keen on that. It's a classical dose escalation study. Focus will be on safety, of course, as this is a first in man, but it is in patients. So we are taking kidneys, donated kidneys, infected with the virus, perfusing them and treating them with SO-002, and then transplanting them into transplant recipients. So safety is key. We're also going to monitor exposure. We expect there will be no exposure. So we have done that also in the preclinical settings, that we treat, we wash, then we have measured, and there's basically no drug left. So we have this situation where we will treat the organ and the recipient will not really be exposed to the drug, something the regulators actually appreciated. And then because we are going into sort of real patients that will get an infected organ, we also have an opportunity for an early efficacy readout in this first study here.
I was going to ask, when do you anticipate? I know you're only just starting, but when do you anticipate being able to get some results?
So result will come on a rolling basis over the coming 10 months. So by the end of the year, I expect us to have the safety data from the cohorts. Moving into the first quarter of 27, we should have some of the maybe early indications of efficacy at that point in time, because we're following the patients for 6 months, looking for viral reactivation. And we know that the background viral reactivation in this cohort should be somewhere between 50 and 80%. So it's actually a quite big window that we can look into with the expected efficacy that we have.
What does it mean for patients ultimately?
Yeah, so I think for patients, this is going to be a very different approach. Hopefully, they will not have to deal with CMV infection after transplantation and the consequences of that. And that is, some of the direct consequences for how well the transplant will actually go with the kidney in this case, but it could also be other organs. They don't have to deal with taking antiviral drugs for 3, 6, 12 months after transplantation, some of them with annoying side effects. So I think it's going to be very significant that you can avoid the risk of this, or lowering significantly. I don't think you can fully avoid it, but lowering significantly the risk associated with CMV and really changing the paradigm of how we treat these patients. And I mean, that would be what we aim sort of ambitiously after, of course, is to change how we treat these patients.
You mentioned earlier about educating people about what you're doing. You were recently at the Cutting Edge of Transplantation Summit and most recently Bio Europe. What were the reaction like?
It doesn't require that much education when we go to the transplant meetings, honestly. They know what they're dealing with. Everybody knows about machine perfusion of organs. Everybody knows about CMV. It has been a significant problem in this space for as long as you have done transplants. So there, I think it is really some interesting discussions we can have. I'm happy that some of the data that I talked about, they have just been out in American Journal of Transplant. There was a very nice editorial on the concept following that just 1 or 2 weeks ago in again, American Journal of Transplantation, highlighting some of the potentials, but also, of course, some of the challenges going forward with this path. So I think that is actually really helpful to have these discussions. From a clinical perspective, I think there is unanimously very big support for doing something like this. It makes sense. So there is quite a strong support. We have good KOLs supporting us both in US and Europe. So I think it's meaningful. It's not only significant, it's very meaningful for them to think about the opportunity to actually eliminate the virus, even before we put the organ into the transplant recipient. That is very meaningful. BioEurope is different. It's not clinicians dealing with this on a daily basis. It is BD from the pharmaceutical industry. We, of course, meet companies that are in the space of transplantation, kidney disease, infectious diseases. And I think we are met with, again, curiosity. This is really a fascinating new treatment paradigm that we are proposing. And I think that is paving the way for some good discussions with them, that we can actually do something novel. Again, novel is good. It's also challenging in some instances. Yes, you need to develop something new that has not been done before. But I think we come with something that is also de-risked at this stage. We have already basically done the phase 1 study, except we didn't transplant the organs, but we have done numerous treatments of human organs with good results and is now moving into the clinic. And I think when we have treated a few patients, I think the mystery sort of will disappear and we will be on a very good path for good discussions at that level too.
Does that mean that you're looking ultimately for partnerships?
I think we're not different on that level from any other biotech company in the world. We are looking for whatever means of financing we can find. That said, I actually think there is value in itself to have early discussions with pharmaceutical companies in the space. And we have nurtured some of these relationships actually over the last few years. So we already have good relationships with several companies in the space here. And I think that brings value to understanding what competences we need, what we need to focus on in our development. And then one day that could potentially lead to some kind of transaction with one of these partners. So it serves the purpose of course, a long-term relationship building, but also, of course, could there be an opportunity for a partnership or down the road a transaction? I think a transaction in this space is likely to happen after our phase 2 program, that we get more solid proof of concept efficacy data. And this is also what we are now looking for financing for. That is, the financing we have right now will take us through the safety study here in the phase 1. And now we are out looking for financing to start these early discussions for financing for the phase 2 program.
Are there any other applications for 002?
Yes, in the sense that kidney transplantation is not the only organ that is affected. You have very similar challenges in liver transplantation. So liver is, of course, on the agenda, and you have even more problems when it comes to lung transplantation. So basically all solid organ transplantations are additional therapeutic opportunities where there is a clinical need to do something. They have the same challenges with the organs. They have the same or more severe challenges with current medications and so forth. It's a very similar paradigm you're talking to when you're talking to liver and lung, etc. So this is definitely areas that we are exploring. Yeah.
And so what are the next steps for the company, I guess, working on the trials?
Yeah, the next step is to get through the phase 1 study here, get the safety data, get the PK data and exposure data, looking at early efficacy signals. It is to get regulatory alignment on the later stage programs, phase 2 and registration studies. So we sort of can home in on that. And then it is to start broadening out into liver indication and lung indication and see when we can actually start those programs. So that will be the next step we are looking for. And then under everything we do, there's always financing activities. The phase 1 study, we were actually working on a different path originally, but then we were approached by UHN Toronto and the PI there, Atul Kumar, and that they would pay for the study. So it's actually a sponsored, investigator-sponsored study that they have initiated in Toronto. We're really pleased about that. They really want to make a change in this space here.
Both interviews today on something truly groundbreaking, and both have the capacity to make a huge positive difference to patients globally. So that's it for this week's podcast. If you do want to get in touch with guest opportunities, or if you want to advertise, please feel free to contact us at jim@deeptechdigest.com. And just that occasional reminder, we don't charge guests to appear. I know there are some podcasts that do. So until next time, wherever in the world you are, I hope you have a great week ahead. Thanks for listening, and I hope you'll join us again next time for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.